Greetings to all the learners. Let us learn about in situ hybridization in this video session. To define in situ hybridization, it is a technique that uses labeled probes to detect specific nucleic acid sequences within intact cells and tissues. And the principle behind it is the designed probes will bind to the complementary target sequences, which reveals the presence and the abundance of the target molecule in the fixed tissues. So the three key steps in this in situ hybridization are probe designing, hybridization, and signal detection. Probes are short single-stranded DNA or RNA sequences complementary to the target sequence. And in hybridization, the probe is incubated with a sample, allowing it to bind to the target sequence via complementary base pairing. And in signal detection, the probe is labeled with a detectable marker such as a fluorescent dye or an enzyme, which enables us to visualize the target sequence on the fixed tissue. Sample preparation includes tissue fixation, embedding and sectioning, and permeabilization. In tissue fixation, the tissues are fixed using formaldehyde or paraformaldehyde to preserve morphology and nucleic acids. The fixed tissues are embedded in paraffin or frozen, and thin sections are prepared on slides. The sample is treated with enzymes or detergents to make the nucleic acids accessible. Probe designing and labeling. The probe must be designed to bind specifically to the target sequence, and the labeling includes it has to be labeled with markers, which can be a fluorescent dye or the enzyme, which enables us to detect and visualize. And the probes can be classified basically into three types depending upon the source, that is the nucleic acid type. Either it can be a DNA or an RNA, which can serve as a source of probe designing. And the synthesis method, it can be either cloned, synthetic, or by using PCR technique. And it can be used for either for repeated sequence, for single copy, for whole chromosome, for part of chromosome, or for total genomic DNA. And the labeling of the probes. Labeling of the probes, the slide shows the non-radioactive labeling method, which is enzymatic and chemical, and the radioactive method of labeling. Under enzymatic, we can use biotin or digioxygenin as the enzymes to label the probes. And under chemical labeling method, acetyl aminofluorine and mercury, that is any metal, can be used for labeling the probes. And the radioactive labeling includes isotopes of sulfur, phosphorus, and hydrogen, and all the beta emitters can be used where it can be detected by autoradiography method. And in this non-radioactive labeling procedure, we have two methods, direct and indirect. Under direct method, the label is incorporated directly into the probe so that the hybridization site could be visualized immediately after hybridization. Whereas in indirect method, the label cannot be detected directly and it requires a reporter conjugated with the signal generating system. And here we have label and reporter molecules. So if we use biotin, avidin or streptavidin should be used as a reporter molecule. And this reporter molecule will be conjugated with the signal generating system. As we say, it is an indirect method of visualization. And if we use digoxygenin, then anti-digoxygenin antibody has to be used. Acetyl aminofluorine, anti-acetyl aminofluorine antibody, mercury, ligands having same immunogenic group which can bind to same antibody will be used as a reporter molecule, and the reporter will be conjugated with the signal generating system. And what is a signal generating system? The signal generating system can be classified into three. The first one is fluorochrome. So the fluorochrome are molecules, they get excited by light of one wavelength and they emit the light of another wavelength, which we can observe as fluorescence of different colors. Enzymes. Enzymes catalyze particular reaction, for example, precipitation. At the site of hybridization, which can be visualized as a visible product. Metals. Colloidal gold is conjugated with antibodies and it can be visualized both by light and electron microscope. Here are some of the color codes of signal generating system. Under fluorochrome, if we use fluorescence isothiocyanate as a signal generating system, it gets excited by the blue light and it emits green fluorescence, which we can visualize at the site of hybridization in the in situ technique. And if we use tetramethyl rhodamine isothiocyanate, which gets excited by the green light and it emits red color light. And Texas red or sulforhodamine, it gets excited by the green light and we can absorb deep red as the fluorescence. Aminomethyl coumarin acetic acid gets excited by the UV light and we can absorb blue light as the fluorescence. And in the case of enzymes, for example, if we use horseradish peroxidase, the substrate will be diaminobenzidine and it catalyzes to get the visible red precipitate which can be absorbed in the slide in the fixed tissues. And in the case of using alkaline phosphatase, the substrate used will be 5-bromo-4-chloro-3-indolyl phosphate. Blue precipitate can be absorbed in the fixed tissues, which shows the site of hybridization. Let us learn the technique using an example, detection by biotin. Suppose now we have a nucleic acid probe which we have designed complementary to our target sequence and we are using biotin attached to this probe. Now we have designed a probe using biotin. We have a single-stranded DNA, that is our target DNA, and now we are allowing the designed probe to hybridize with the target sequence which is labeled with the biotin. Now after incubation we have a hybrid DNA. This hybrid DNA has to be visualized by this indirect method where we are using biotin as a detector compound. Avidin, which is a reporter molecule, is synthesized and it gets conjugated to the biotin. Now this avidin is conjugated with the signal generating system, that is FITC. So after the incubation, the avidin goes and binds to the biotin and it emits green color fluorescence which we can absorb in this hybridization site. By this way, we can locate the presence and also the position of the target sequence in the fixed tissue sample. Getting into the applications of in situ hybridization, this in situ hybridization technique has a wide application in the areas of gene expression analysis, chromosome mapping, disease diagnosis, developmental biology, and cancer research. And the advantages and limitations of the technique. Advantages, it gives a high spatial resolution and it is a very versatile technique with a wide range of applications. And the limitations include specialized expertise and equipment for probe designing, sample preparation and signal detection. And also it shows a limited sensitivity and may not be detectable when there is low levels of target sequences depending on the chosen probe and detection method. Thank you all.